Namaskar, my dear friend. You again welcome to my YouTube channel DP Forensic Lab. Today video I will discuss about a very useful and advanced technique PyGCMS, pyrolysis gas chromatography mass spectrophotometry. This is a very useful and advanced technique. I will discuss about its application in this video. So first, what is a PyGCMS? So as the name is indicate, it is a PyGCMS. GC, you know that the GC gas chromatography, MS mass spectrometer, and this attached with the pyrolysis. One pyrolyzer is attached. This is a GC, this is a MS. MS is attached with the GC, and initially this start from the pyrolysis, and this is a computer system is operate in the all system. So I will discuss about this technique. PyGCMS is a very useful technique. So what is a PyGCMS? This is a combination of the four techniques: pyrolyzer, GC gas chromatography, and mass spectrometry. So what is a pyrolyzer? In the pyrolyzer, sample is rapidly heated. The sample is rapidly heated to the high temperature. In the chamber, the oxygen-free environment, causing it is a breakdown into the smaller volatile fragment. This process is called the thermal degradation or the pyrolysis. Now, next is attached with the GC. The volatile or fragmented of the samples which fragmented by the pyrolyzer. Pyrolyzer break down the samples by heating at high temperature oxygen free environment so this volatile fragment are swept in a carrier gas into a GC column. Here they are separated into individual component, as we know that, based on their different boiling point and polarities. And in MS mass spectrometer, each fragmented compound enter in a mass spectrometer where it is ionized and fragmented. The mass spectrometer then measure the mass to charge ratio M by Z, and resulting the fragment creating a unique mass spectrum. Creating a unique mass spectrum that serve a fingerprint in the identification. So this technique, three techniques are combined together, and this will give a beautiful result. PyGCMS. This is very useful techniques and can be used for identification of an organic compound, different type of organic compound. So in the forensic lab, it is very useful equipment or the technique. This can be used for the paint analysis, different type of the paint. Here fiber, different of the fiber, synthetic fiber give the good result, different of the synthetic fiber, ink and toner, adhesives, laser toner, cosmetics, different of the cosmetics, because cosmetics contain the organic compounds, organic substances, lubricant, polymer, rubber, DNA, or any etc. Any type of the material which contain the organic material. And as we know that in the present era, everything is contain the organic compounds, synthetic compound. So this is very useful techniques. For the reference, this is used in the paint analysis. This is paint analysis. This can be used in the ink analysis. This can be used as a textile fiber. This is a fiber, this is a rubber, etc. So any type of the organic material or exhibit can be analyzed using this technique. So here PyGCMS versus FTIR. This PyGCMS is slightly greater useful or very informative techniques. In case of FTIR, we know that FTIR is a good techniques, but in some cases if the FTIR spectra is not able to distinguish between the two different kind of the samples, the PyGCMS is a very sensitive technique. This can be distinguish the two samples which are very similar in the chemical manner. In where the FTIR is not able to distinguish. So for the reference, this is a reference. Different similar kind of the paint having the identical similar infrared spectra. If the IR spectra is very similar due to the chemical nature, in that case the PyGCMS can distinguish. So in this case of the paint sample, discriminate among the 25 sample FTIR group the PyGC analysis. Automotive coat in vehicle paint can be used. PyGCMS with the multi-level statistics for the forensic discrimination automotive paint. So this is a very this is useful where the FTIR is not able to identify or distinguish of the two samples. This PyGCMS can distinguish in case of the FTIR not able to distinguish of the two samples. So this is a basic, actually this is a very important techniques. The detail of the analysis, because in our India this presently not available in the I think there is no this equipment not available, and in Chandigarh this is available when I was in that lab. Okay. So but this is a very useful technique, and I think now the other laboratories or the university may have to this type of the facility. What this is important technique or the for the analysis of the forensic samples as well for the research purpose. So thank you. I will make another video for the whole detail about this techniques. But in this video, I have tried to explain the utility of this video, this equipment PyGCMS.